John, thank you very much for inviting me. John and Jane, thank you for organizing, and the other organizers. This has been a terrific meeting so far. Lots of great ideas, lots of great discussion. I want to turn the focus, I think picking up with some of the things that Lee Miller was getting into and Eb Fetz last night. I want to talk about the use of brain-computer interfaces for something that's distinctively off-label. I want to talk about the basic neuroscience applications of this really powerful tool. Okay, so the big picture are the following 2 points. Brain-computer interfaces allow us to ask old questions in a new way, okay? And they also allow us to take a technology that's been with us since at least Eb Fetz, and Eb acknowledged the work of Jim Olds 10 years before him, okay? And we can use these techniques to open up some new questions in neuroscience. So you all know what a brain-computer interface is. My particular angle on it, and the work of many people whose work I'll discuss today, is to use an invasive technique, the Blackrock array, in the brain of an awake behaving monkey and allow the animal to see some transformation of his neural activity on a screen, okay? Eb Fetz recognized the power of that technique over 50 years ago, and it's a rather humbling question to ask how much progress we've made. But bear with me, and I'll try to make the point that we've been able to learn a lot about how the brain controls movement and learns by using this technique. Okay, so first, just to, I feel like this is a great opportunity for me to represent a community of researchers, and in order to do that as well as possible, I'm going to let the work of Vikash Gilja and Paul, how do you say his last name? Paul Nyukian, who's now a faculty member at Stanford, out of Krishna Shenoy's lab. This is a video that represents the state of the art of what we can achieve with invasive technologies and sophisticated decode algorithm design. That's fantastic. Lee also referenced this paper from Steve Chase, Byron Yu, Matt Golub, Andy Schwartz at the University of Pittsburgh. Their particular focus wasn't just on good BCI performance, it was also on being able to stop a cursor and hold it there. Like Lee Miller mentioned, postural control in M1 is still quite an enigma, okay? But they were able to use a cognitive trick that the animal could harness to stop the cursor and hold it still. So this is the state of the art in our field right now. Having great performance like that allows us to keep careful tabs on the neural correlate of behavior, and a BCI makes that possible. So how do we use a BCI for basic neuroscience, and what advantages might it have? So first, when you're using a BCI, you have a bottleneck between the massive billions of neurons processing in the brain and a behavior, okay? In our case, the behavior is the cursor of a movement on a screen, but you can imagine a high degree of freedom control with a robot arm, okay? We've got all the neurons that directly drive behavior under our control, under our observation, okay? You can also decide to leave out some neurons. You can watch what the neurons that directly drive behavior do, and you can observe what the other neurons that are along for the ride do at the same time, okay? We can design the decoder, understanding how motor cortex controls the native arm is a huge, wonderful, and complex problem. I hope some of what we do with a BCI can turn around and inform those efforts. Okay, but for us, we can give the animal a simple, linear, low-dimensional mapping that we know entirely and study whether the animal can learn how to control a cursor under that mapping. And then we can change that mapping and induce learning and understand how neurons change as a population to control a new device. Okay, as Lee alluded to as well, feedback is relatively under our control by asking the animal to, or the human being in a similar paradigm, to hold their arm still. We can actually eliminate proprioceptive confounds on neural activity and simply take control of the feedback by limiting it to the visual domain. Okay, I'm going to talk a little bit about how we can harness these advantages to learn about motor processing and cognitive function using a BCI approach. And These are the questions that I think motivate many of us in this field. I don't have direct answers to any of these questions, unfortunately, but I think we're beginning to get some hints in the studies that I'll survey for you. So first, what are the brain's mechanisms of skill learning? How is it that the ability to play piano is mapped onto synaptic connectivity? What brain areas are involved? Plasticity versus flexibility. When can we say that synapses changed? Let's hold that out as a high bar for plasticity versus mere flexibility, learning how to cognitively cycle through behavioral strategies to work with a new effector. We spent 40 years, as Eb mentioned yesterday, conducting individual neuron studies. Okay? Only over the last 10 years have we had the ability to look at populations of neurons and the networks that they instantiate. How do individual neuron tuning properties relate to an entire network of neurons? Is it one-to-one? Is it obvious, or are there more subtle interactions? Okay? How much of an impact does context have on neural function? So there's going to be a great workshop, I think it's tomorrow, about whether, I think it's a workshop, whether we should train a BCI using imagined movements or attempted movements. Those drive M1 differently, we believe. Okay? And I think that the animal studies can give us some sense for how M1 goes between native arm control, BCI control, multiple mappings in a BCI. Okay? These are questions that are within reach for us. What's the input-output mapping of a cortical area? I'll mention quickly later on, but I want to make the point now that encoding is not the same as decoding. Okay, what you get when you're measuring tuning curves of individual neurons in M1 is not as what M1 is controlling downstream. So how can we understand how an area, a circuit of neurons, can take an input, map it to an output? Okay, finally, what are the units of volitional control? Okay, Eb was getting into this with his talk yesterday when he was talking about the dissociation paradigms that he used. When we are asked to volitionally move a cursor on a screen, at what level is the nervous system changing? Is it single cells, columns, large-scale networks spanning multiple areas, some frequency-based signal? What is the unit of volitional control? Okay. If we want to do BCIs for basic neuroscience, there are two powerful ingredients that we can use. One is neurofeedback, okay, letting the user, human or animal, see their neural states through some transformation that hopefully doesn't corrupt or add too much to the neural signal and gets at the core of what the information the animal is controlling actually is. And then new concepts from computer science, machine learning, and classical statistics related to dimensionality reduction. These are two very powerful tools that enable many of the studies I'll show you. Okay, acknowledgements before I get into the meat of things. So these are ideas that have been developed in large part with Byron Yu, my collaborator, Steve Chase, another collaborator, and Matt Golub, who just earned his PhD, and also conversations over the years with Lee Miller, Krishna, John, Jose, Carmina, Rui Costa, etc. So I'm really representing, I hope, a broad range of different people's work, and I hope I do the work justice. Some of them are in the audience. If I don't represent your ideas well, let me know, question and answer, or afterward. Okay, and then when I get into talking about work from my lab in collaboration with Byron and Steve, these are the people who did the work. Emily Obi is doing follow-up work that I'll discuss. Alan Degenhardt, another postdoc in the lab, is talking about his PhD research Thursday afternoon. So, and this is all just would not be possible, wouldn't even be conceivable without funding from the NIH. Okay, so history and then recent studies, finally where we could go from here. You saw this yesterday. If you haven't read this paper, go read it, okay? It is amazing to realize how much of what we're dealing with now Eb was already thinking about nearly 50 years ago. Okay, in this simple paradigm, Eb was able to condition monkeys to volitionally modulate the activity of single neurons in M1 up or down in order to receive food pellet or some applesauce. Okay, Eb gives credence to an earlier study that did the very similar approach using individual motor units in a human. Okay, and Jim Olds apparently was doing it back in 1963, which is awesome. Okay, great. This is another early progenitor to our field. These are the papers that we look back on as having been the origin of of at least invasive BCI approaches. Okay. I realize the non-invasive approaches may trace a different legacy, which may go back even further, but these are the papers that we read and reread and feel humbled by. Okay. Don Humphrey was able to do an offline reconstruction of movement parameters, movement of the wrist, based on a population of 4 or 5 motor cortex neurons. Okay, take a moment to read this. I'm not going to read it out loud, but can everyone in the back see this? Put up your hand if you can actually read the quote. Okay, thank you. Just take a moment and read it. This is in the footnote of this Donald Humphrey paper. Using such signals for the control of a prosthetic device becomes a less remote and a more interesting possibility. And then he traces the lineage even further back than that. Here we are, 50 years later. What do we got? Ebb showed this yesterday. He didn't show that one. He showed this one. This is a pair of neurons jointly co-modulated, either up together, down together, or actually up and down. Okay? This is the shining example. Ebb was able to try this 4 or 5 times. And I think if he's here, he can tell me if this is true or not. I think this was the most compelling example of this experiment. The thing that they did was I think this might still be he, no, this is with Marianne Baker, monitor another neuron that was not part of the conditioning, and that neuron tracked the neuron that was being conditioned. Okay, so more recently, sticking with pairs of neurons, Anderson did a very nice study in which they made the point that BCI-induced plasticity, or changes in neural function, not plasticity, changes in neural function, correspond to the animal's natural movement repertoire. So they had an animal perform a real reach, and then they had a BCI task, left or right, using a single neuron. And then they had the animal switch the contingency, so now it's an anti-BCI movement. Cursor appears on the left. You have to intend a movement to the right in order to drive the cursor to the left. It's a clever disassociation that had been used in hand control before, but not in BCI control knowledge. And and then this is work done in parietal cortex, so maybe you expect something slightly different from M1. But what they found is that the neuron, the trained neuron, the one for which the animal was receiving BCI-based feedback, had the same response during reaching and BMI pro, where they do the switch task. Okay? And then when the animal is doing the BMI anti-task, this neuron goes in the opposite direction as you'd expect for good control. What was very interesting is that an untrained neuron, let's see, that's the red one here, an untrained neuron maintained correlation with the trained neuron, okay, up or down, down or up, as the animal was doing this task. So Richard interpreted that and Yunjung interpreted that as parietal cortex caring about movement goals, okay, and using the natural repertoire to achieve a new movement. Mark Schieber, who's here, Mark is talking tomorrow, Thursday. I have the calendar at the end. Mark Schieber harnessed the activity of 4 neurons to do BCI control in an up-down task. So the cursor is moving up or down, and you can see vertical cursor position read out from these 4 neurons. And after about 300 trials, the animal was able to achieve a very narrow target band. So the animal was able to get very precise control of the position of that BCI cursor. And that's a learning effect. It took the animal 20 or so minutes to hit that level of performance, okay, using forms. And now Mark has a tool that he can use to study what is it about a population of neurons that allows them to learn at that level of performance. So better performance is down in these axes, okay? So if the animal's doing really well, the size of the target, the RTS, the relative target size, is quite small, okay? You can see that with 4 neurons, the animal does better on average than he does with just a single neuron. But there are some very interesting outliers. Here are some cases with 4 neurons that do worse than many of the cases with 1 neuron, okay? Mark also examined whether the native direction tuning of the neurons was correlated with how well the animal could harness them to do this task, and that relationship is basically flat. He also examined the native tuning not just in terms of preferred directions, but in terms of shoulder or wrist activation, And the native proximal distal properties was not a strong predictor of how much learning the ensemble could perform. So, yes, Mark's speaking tomorrow. He's our kickoff speaker in the Triron room, I think. Okay. Jose Carmina, working with Karanesh Ganguly, gave an animal a scrambled BCI decoder. Okay. They took all the weights from a good baseline decoder and they scrambled them. So the animal had to learn how to control this scrambled effector. And it took the animal 3 days to get remotely good at it. And then whatever he was doing stabilized and performance remained high as long as they could hold the 12 or so neurons involved in this paradigm. Okay, so we've got, let me say one more thing about a different paper from Karanesh and Jose. They then gave animals 2 different decoders and asked the animal to switch between them. And he could do that after some initial learning. Here's day 1. After some initial learning, he could switch no problem. So there's some context dependency to what M1 is doing in BCI control. It's hard to get really different movement contexts if you're using your natural effectors. Okay? And then they could go a step deeper and look at the native tuning of these neurons under the 2 different mappings. 3 individual neurons. Some have different tuning, different in terms of modulation depth. Others have different tuning in terms of preferred direction. Some neurons do the same thing under both maps. Okay, and then Karanesh is speaking also tomorrow at 11:25. Okay, so here's a quick, I know I'm going very quickly, but I really want you to kind of get a survey of what's been done in the field of BCI learning, and then I want to try to go on from there to work from my own lab. But let me just summarize what we've shown so far, what I've talked about so far, and what many other wonderful papers have shown. Fett showed us that correlations between some pairs of neurons can be modulated training. Anderson showed that neurons preserve their correlations even when the animal is doing a novel BCI task involving an anti-reach. Okay. Anderson would make the argument that BCI learning is tightly linked to the native tuning. In Mark's data, that is not the case. BCI control has very little direct relationship to the native tuning of the neurons involved. Okay. Mark's learning was rather rapid, converging over 20 or 30 minutes. Jose and Carmina were able to, Karanesh and Jose were able to design some decoders that took the animal upwards of 3 days to learn how to control. How do we reconcile all of these disparate findings? Okay, so this is work by Patrick Sattler, his PhD thesis project, and Byron Yu and I jointly advised Patrick on this project. Okay, this is a little fanciful illustration of the ideas that I want to describe. Okay, so are using the standard Blackrock array. We're recording the activity of 90 neurons. The animal is controlling a BCI in a 2-dimensional space. Okay, but I'm going to simplify everything in order to give you some geometrical pictures. So imagine we're recording just 3 neurons, and imagine the animal's task is simplified down to 1 dimension of control, not what the animal ever actually did, but the intuitions will be the same. Okay. The other intuition I want to harness is by thinking of a decoder as a mapping, a projection from one space into another space, because armed with that thinking about things, a lot of manipulations become self-evident that are harder to think of when you're dealing with a long equation with many coefficients in it. Okay. So in the simplified example where we're recording from 3 neurons, you can measure the activity of those 3 neurons, just count the number of spikes, okay, and represent that, that's in a 50 millisecond window, represent that as a point in the joint firing rate space. Lots of people have done this before us. I hope many go on to do it because it's a really powerful technique. But here's the setup in case you haven't seen it before. We can move this window through time across an entire session and get a population scatter plot of neural activity. Okay, so that's the neural state space. Now, what happens if you calibrate a decoder? We take a highly skilled BCI monkey. All we need to do is drift the cursor to the right or to the left. I don't know if he's attempting movements or if he's not actually moving. We know that, but he may still be attempting movements. It may be motor mirror neuron type effects. We're not really sure what's going on, but we know that neurons fire in a very consistent way as the cursor moves to the right and fire in a very different way as the cursor moves to the left. So we can use that to fit the line or plane of best fit and we get the mapping from neural activity to behavior. So points that are near this end of the line project onto the line and cause the cursor to move fast to the right. Okay? Points that are down here are going to cause the cursor to move fast to the left. So what happens if you rotate that control space? The animal has to try to figure out what he needs to do to restore control of the cursor, because these neural activity patterns which used to move the cursor to the right are now moving it to the left. And the neural activity patterns that move the cursor to the right aren't really ones that he was expressing before that manipulation. So can he figure out how to do this? And then just to kind of put it on a little bit of a general statement, BCIs allow us to request new patterns of neural activity and observe whether the user can generate them. Okay, so these data are clearly correlated. We use factor analysis, which is better suited for neural data than principal components analysis, in order to find a plane or a hyperplane of best fit to the data. Okay, and this type of dimensionality reduction technique is common, but we just gave it a name. We call it the intrinsic manifold, and we think it's telling us something about the native properties of motor cortex. Okay, so now there are three spaces. There's the natural neural space where you record the data. There's this intrinsic manifold, which in our hands is a 10-dimensional subspace that accounts for most of the neural data the animal exhibits. And then there's a B mapping, which I'm drawing as a line, but in reality, it's a 2-dimensional space that is somewhere within this 10-dimensional native space until we rotate it. Okay, so are some BCI mappings easier to learn than others? Our hypothesis is the following. If we give the animal a new mapping from neural activity to behavior that is within the intrinsic manifold, it should be easy for the animal to learn how to control that. If we give the animal a new mapping that is outside of the intrinsic manifold, that should not be as readily learned. That is the pretend. Let me quickly show you the behavioral result, and I'll show you a video. Soon after the perturbation, performance is reduced dramatically. It takes a while to come back up, 10, 20 minutes. Comes back up. This is for within manifold perturbation, one of the ones that respects the natural properties of the population of M1 neurons. Okay. Back to the original mapping, which we had designed to be as easy to use as possible. There's an after effect. Okay. If we give the animal an outside manifold perturbation, performance is impaired and it stays low throughout the entire session. For most of these, there's very little learning, even though all the animal has to do is figure out what he needs to do with his neural activity to move the cursor well in 2-dimensional space, which he had no problem doing before the perturbation. When we take the perturbation away, he picks up where he left off. There's no after effect, which is a hallmark that there was no actual learning. Okay. So here's some cursor performance. This is not, systems haven't been designed as meticulously as Vikash and Steve and others have done. So performance is not quite at their level, but it's good enough for us to be able to create manipulations. Here's what happens after one of these perturbations. The mapping from neural activity to cursor movement has been disrupted, and the animal cannot harness good control of the cursor. Here's what happens after about half an hour. With the within manifold perturbation, we did many of these, but this is an example of one of them. Cursor performance is pretty much back where it should be. Okay. Outside manifold perturbation, cursor remains impaired even an hour after exposure. The animal keeps working away, not realizing that we've predicted there's going to be no hope of him getting good cursor control. So why is it that this BCI mapping is so difficult for the animal to use? And are there lessons from this about how the brain processes movement natively and how we can design better clinical BCIs? Okay. So let me sum up what I think this study shows us and then just take a couple of minutes at the end to talk about future directions and what other people are doing with the BCI approach. So this concept, intrinsic manifold, I think unites disparate observations. It helps us understand why some BCI mappings are fast to learn, others are slow to learn, okay? Simply because some new BCI mappings respect the existing topology of population patterns, others violate it, okay? Now we have a tool that allows us to see that and to design decoders that we expect to be easy to learn or more difficult to learn. Okay? That's, people have struggled to find clear paradigms, especially that we can use repeatedly in monkeys that harness new, that engage new learning. And in a BCI approach, we can do that. So we've taken the simple thing to do to study how learning is controlled, and we've made it easy simply by doing dimensionality reduction and giving the animal an effector that lies outside of the low dimensional space. Okay? So there are a lot of studies of dimensionality reduction, and I think that this type of study says that all of those other studies are something real about how populations of neurons behave. Okay? It's also very fun to think about what you can do if you have some question you want to know about neural processing. Can you reframe it in a BCI framework and maybe say something new about it? So Eb talked about the idea of disassociating cortical neurons from EMG activity. Whatever your thing 1 and thing 2 are, you can test if they're disassociable by putting them in this competitive space. Okay. So I want to manage my time well because I'm getting signals that I'm cutting into my question time. So I'm going to, if anyone wants these slides, just grab me, or if you want to talk more about these, by all means, I would certainly love to. I'm going to crank faster than they deserve through a couple of really cool things so that I can make a point at the end. So this is great work from Krishna Shenoy and Matt Kaufman and colleagues showing that the concept of a null space applies during real movements as well as during BCI call. Okay? Mark Slutsky and Mark Sieber have both looked at very similar things in recent papers, worth looking at. Okay? There's a debate about what the native function of M1 is. Okay? We have this old model from Georgopoulos' early studies in the 80s that M1 is cosine tuned. More recent work from Krishna Shenoy, again with Mark Churchland and John Cunningham, showing that populations of M1 neurons seem to instantiate a dynamic system. If nothing else, this makes the point that M1 encoding and decoding are fundamentally different processes in native arm movement control. Okay. Tieran Moore did some Fetzian modulation of frontal eye field neurons. Can we make this a word now? Fetzian. Originally it said volitional, but let's go with Fetzian. Fetzian modulation of V1 neurons, of frontal eye field neurons, affected visual attention, but not eye movements, suggesting that those are dissociable processes. Okay? Karnesh Ganguly can speak for himself because he's talking in a workshop tomorrow morning. So please come learn about how BCI consolidation can be studied through sleep mechanisms. Okay. Ending with one more set of very intriguing studies. Christoph Koch, Itzhak Fried, and their colleagues conducted medial temporal lobe recordings, isolated the tuning to visual images, faces mostly, of 4 neurons, and then they allowed patients undergoing epilepsy surgery to volitionally modulate what was presented in front of them on a display. And people could usually fade in a picture of Marilyn Monroe or Michael Jackson, depending on which neuron they were asked to volitionally modulate. That's a cool study, but one of my favorite things about it that I think points toward the future is that they asked people what they thought they were doing. That's a whole different type of data. This is data that we cannot get with our monkeys. We can't ask them what they thought they were doing when they perform BCI manipulations, but with a human you can, so why don't we? Richard Anderson, a very nice study doing parietal cortex BCI control, but when you read this paper, one thing that really stands to me is the idea that the BCI user himself seems to have been a partner in the research. There are verbal descriptions in the paper of the BCI user finding that he could activate neurons by thinking about touching his nose. And I don't know if that's the type of thing that we could ever actually get our monkeys to agree to. So why don't we continue to do what these pioneering groups have done, and many other people have as well? I'm taking a very selective look through the literature. Why don't we incorporate verbal reports in our scientific data? Okay. So I'm going to gloss through the future stuff. There are challenges. I have a bit of a response to John, which I will give to him personally because I think it's worth getting clear on what exactly we're studying with BCI control. So I'll just leave it now as a challenge for the field. Okay. There are also opportunities to invent new things we can ask about neural function through a BCI approach. Okay. I want to say that we should consider very seriously taking verbal reports as data, as long as we're cautious about it, okay? You don't want to bake in the result that you get back out, so you need to do your best to use double-blind methodology, okay? But what if we go further, okay, a little bit further into the future? Think about first-person perspectives as something that we might want to incorporate in our data sets. Okay, another rule of thumb, I would not want to read a paper that says, I observed this in my fancy high-resolution fMRI machine, and therefore that's the neural basis of vision or perception you want to say. But maybe first-person reports could be useful to setting new hypotheses which can then be held up to the standards of objective, verifiable science. Okay, and I want to draw attention to this nice review article that brings up some very intriguing ideas along these lines. Okay, so if we were to take a first-person perspective in some BCI of the distant future, what types of things could we do? And my last question of all is, when do we get to have our consciousness machine? When can we go into a high-resolution type of scanner that's giving me single neuron resolution, the entire brain, and allowing me, as an experienced neuroscientist, to concoct new hypotheses about what parts of the brain might be involved in perception, imagery, and volition. Okay? Maybe this is something that we can collectively drive for. Okay? As long as we're careful about good scientific methodology, we don't fall into philosophical pitfalls along the way. Okay, so BCIs are an older, the neurofeedback approach has been with us for a very long time. We can ask new questions with it. It's something that I think many of you who have access to human beings can begin to do right away, and it's something that we just kind of harness with our monkeys, okay? And I gave my talk a little slogan, which is BCI for basic neurosci, and you can too. Okay. These are our workshops tomorrow. If I have any time left at all, I'll take questions. If not, I'd love to talk throughout the meeting. Thank you.